then we pick the street name.
I think so. It really, and then normalization, so the normalized n is
It's like use down links that are in the region. You're selecting
normalization. Like he put no here because he didn't solve some there's different ways to normalize it, but the convention
So it's kind of like
it's all real. Yeah. That's how you point.
how many are at that point is divided by 1,000.
No, that's the first and then the idea is use those as a coefficient.
Do we just select by location? Yes. Yes, select by location.
That's the one I started, but
No, this is what I
Not wilderness.
is just converted to refrigeration form. So what I did was just expanded the size and then converted. And so she mapped this first. Like you took those as buffers together.
I have been writing it this way, but I need to change it to the rate of change.
It's the very first thing.
I don't know which way to write it. I'm pretty sure I could here because
We don't have a flag
to
search.
try to map because the expression's right. And you have to find the expression making
Last time we applied to.
if you want to be sure. So anyways, that's what I did.
Okay.
So then go.
How do you make it discrete?
You go
We get a function here.
to within this
A sum of the but yeah, I suppose you can do it either way. Because it's just this part at the end where it's
just like press the
calling
for itself.
So do it either way you want. I mean, mine turned out.
We're in the selection.
These two ones.
Taking 99 data. Oh, so what time?
So today we're going to start discussing the kinetic Monte Carlo model that's used to solve the affinity discrimination problem in B cells. And the project problem you're going to do is based on these two things. So I'll post the project problem next week on Smart Site. So you'll do a kinetic Monte Carlo simulation of its kind of very simplified description of B cell signaling or B cell receptor ligand binding. So today what I'm going to do briefly again touch the topic of affinity discrimination in B cells and start discussing kinetic Monte Carlo for 1 hour, and then we'll have a help hour. The next 1 hour, I'll have help hour for more problem. So if you remember, I discussed in earlier that when B cells bind ligand molecules and degenerate. This receptor ligand binding you can describe by 2 parameters. One is how strongly they associate. That means they form the complex when they are in free form, or how they dissociate. If you are in a complex form, how you dissociate into free receptor and free light. Now the ratio of these two is what I think. That can vary over a wide range in B cells. That means it's changes over 5 orders of magnitude, which is a huge range. But if I plot the signaling, shows behavior like this. Okay. And you know what these different curves are, like what I'm varying here.
Steady state concentration.
No, this is over time. So x-axis is time, and y-axis I'm plotting some measure of signaling, and I'm going to be more specific soon. But let's say some measure of signaling over time. But I have a bunch of different curves, and what I'm varying, like what's changing. Okay. So I'm changing A. So this you can think of like 10 to the power 5, and this top curve like 10 to the power 10. You see that as you increase in affinity, it keeps increasing. Now, if I want to plot the signaling at any time point, even before reaching the steady state or once in steady state, three, and if I plot that signaling this is KA, and this is how does this look? Okay. So it's going to look like so there are two ways you can plot this signaling graph. One is you can plot the time course, and you vary affinity for different like Ka values. You can plot this curve, or you can fix time. So at a fixed time t equals t1, you can vary Ka and plot signaling. Now this is the behavior you see for B cells. Now do you recall I discussed this a little bit? If I do the same thing for T cells, y-axis is signaling. Do you remember the curve? Right, it's a non-monotonic curve, so that means it goes up, but then it decreases. So there is So that I bring that result or bring that analogy here because it tells you that naively one might expect that okay, I increase affinity and the signaling should go up, but it should not be true always. So that means there is more to this problem than one might have thought in a naive way. So if you plot the same type of curves for 3 cells, you will see that it goes up, 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 and then later affinity values you will see the graphs are going down if you plot the time course. Then the question we ask, B cells. So what we are trying to do, that we see this kind of activation, but can we understand that this behavior in terms of elementary reactions, meaning receptor ligand binding and subsequent signaling reactions. So can we create a simple model with major membrane proximal signaling events, and can that provide insight into this affinity dependent signaling? So that's kind of what we are after. And we are going to describe. Let me describe a very simple network model. They can bind these ligands, and this is also on the antigens are also on another cell surface or on a bilayer. And then I will have two molecules. One is BIM molecule, and then there is a molecule called SIG. So these are two major signaling molecules close to the membrane. So what do I mean by membrane proximal? Meaning this receptor ligand binding is happening at the cell surface, the B cell surface. And immediately after this binding, there are two molecules, lin and sig, that kind of propagates the signal. Once sig gets activated, then it kind of takes it downstream inside the cell. So, but we stop here at the level of SIG, and we show that whatever discrimination you see at the level of SIG is going to get propagated downstream. It might get amplified through feedback loops, positive and negative feedback loops, but eventually you are going to see similar qualitative behavior. Meaning, if you have this kind of B cell type graded response, like you increase Ka, signaling keeps increasing, versus T cells, it goes up then goes down. So these are two kind of very different qualitative behavior, and that happens already at the level of SIG. After that, you cannot go from this kind of graded behavior to this, or if you have this behavior, you cannot go to that. So this like very fundamental qualitative difference that's going to propagate from SIG. Okay? So that's why we focus on the very first event of membrane proximal SIG. Now, how lean and SIG, what they do? So lean So there are two-step phosphorylation reaction. The first step is lean can bind with BCRs, and each BCR molecule, so these are BCR molecules, each BCR molecule has two sites, which we call Ig alpha. So those are two molecules attached to the cytoplasmic chain of BCR, okay? And those two molecules get phosphorylated by lean molecules. So lean, and do you know what phosphorylation means? It's attaching a phosphate group to that molecule. Okay? So lean attaches a phosphate group to BCR, and once it attaches a phosphate group to BCR, BCR means Ig alpha to beta, then SIG binds to it and gets the phosphate group. Okay, now if I ask you this question, why does it need such complicated BCR mediated interactions? Why doesn't lean directly phosphorylate SIG? What would happen then? What would be the problem? Suppose lean just phosphorylates SIG. What would be the problem?
Signaling all the time.
Right. So it's going to, if you somehow allow that lean can phosphorylate SIG, you are going to get activation of B cells all the time. So no matter whether you have foreign antigens or not. Okay. And you don't want that. You want like immune response only when you have some infection. Okay. Even if you assume this, that lean phosphorylates BCRs and BCR phosphorylates SIG, how can you prevent that in normal cells? Like even that can happen in normal cells without infection. Of course, if you have lean directly phosphorylated SIG, that's going to happen all the time. But even lean phosphorylating BCRs and BCR phosphorylating SIG, how can you prevent that without any external perturbation?
You could require that there has to be a collective signal on the groups, right?
Yeah, but you need to define what you mean by collective signal. So you see, somehow you have to have this process. Like this process can happen only when you have ligand binding. Does it make sense? Otherwise, like you will get all these lean phosphorylating BCRs and BCR phosphorylating SIG, and you get SIG. But you don't want that. In resting cells, you don't want unwanted activation. Okay. So that means in resting cells, lean cannot phosphorylate BCRs. but only ligand binding can phosphorylate BCRs. So when you create a model, like a simplified model, so those are the considerations you need to keep in mind. So when you are creating model, you need to make sure to generate rules, for example, which will prevent this cell to getting activated in resting state. But it will get activated when you have ligand binding. Now in reality, the way it's probably happening is through rafts. Are you familiar with this concept of rafts? So rafts are small membrane domains. It's on So on B cell, you can imagine you have these shaded regions and small domains. And so far it's known that Lin molecules are mostly concentrated on these domains, and BCR molecules are mostly outside these domains. Okay, so you create like a partition on the cell surface. So then that partition, like if Lins are concentrated some other place, but BCRs are not there, that means BCR and Lin cannot really come in contact. That's one possible way of thinking about it. Of course, there are other details. Like there is a molecule called CD45 that needs to phosphorylate Lin first, but I'm not going to go into that detail. But let's think about Lins are partitioned in some other small domains where BCRs are not. That creates a separation. But once BCR binds with ligand, you create a new types of organization on the cell surface. And there you have BCR and Lin together. And so this is
what happened again? What happened there? Like the Lin and the BCRs together.
right.
Ligand binding.
So once these B cell receptors bind antigen, then the organization on the surface changes. So you kind of reorganize. So you create new domains, and in these new domains, you have both BCR and Lin together. So that means now BCR and Lin can come together. And the formation of these microclusters where you have both BCR and Lin, we think that's also to some extent lipidated or largely lipidated by lipid molecules. So currently my lab is developing a detailed model where I have this formation of BCR microclusters with lipids, and eventually we are going to put Lin into it. But that's a much more detailed model, and it's quite challenging computationally to develop such a model, because you know that there are lots of lipid molecules. So right now we are simulating the order of 10 to the power 4 lipids. and it takes a lot of time. And also, we need to use an energy-based algorithm because the way the interaction occurs, that the BCR lipid attraction changes upon ligand binding. So we're going to come back to this later and show where we use this energy-based algorithm, like Metropolis or the one I was hinting at in the last lecture. Okay. So when we come back to this later, I will describe where we need such energy-based Monte Carlo algorithm. But suppose we don't want to create such very detailed model first. We want a more simplified model which can already provide insight into the process. What will be the assumption? So this kind of shows you when you are trying to model a very complex biological process, you can model it at various levels. You can create a very detailed model, or you can create a simplified version, but it can still provide physical insight. So our starting point was to create a very simplified model, but which can still show you how you can go from like this behavior versus that. It can tell you when you see this kind of behavior versus when you don't see this kind of behavior. Now, if you want to create such simple models, what will be the assumption? Suppose I want you to develop a model of B cell signaling, which will show you this behavior. So what are the things you want to see from this model? You want one, no activation in resting cells. 2, activation upon ligation, meaning ligand binding, and 3, graded response. Okay. So first thing is there is no activation when you're in resting cell, then there is activation when you bind ligand. And the activation is graded. Do you follow this third point? What is graded? Graded means this behavior. This is graded. Means you change Ka and signaling goes up. Versus this is not graded. This is non-monotonic. Okay. So this is I call a graded response or here it's so these are the 3 things I want as outcome, and especially to see when it's going to be graded and when it's not going to be not graded. So that should come out as we vary some parameters in the model. Now let's focus on the first 2 aspects. No activation when you are in resting cells and activation upon ligation. So can you think of some simple rules, not going into these details of membrane, lipid clustering and all sorts of things. Just can I have a simple model, BCR antigen binding, and then lean and seek activation, or lean activating BCR and BCR activating seek. So I have only 3 interactions. One interaction is BCR antigen, then lean phosphorylating BCR, BCR phosphorylating seek. So now can you think of some rules, like what will be the rules of engagement, rules of signaling?
What's the this? Yeah.
Ig alpha and Ig beta. So those are 2 molecules attached to BCRs, but you can now forget about that. You can think that somehow lin can phosphorylate the cytoplasmic side of the BCR, so it can attach a phosphate group. And once it attaches a phosphate group, that can be transferred to C. why don't you think about this for a few minutes, and then I can discuss.
When you have these cytoplasmic proteins, like when you're sick, you assume that they're one mix, so you don't have to track where they are spatially?
No, we do track them spatially.
So is it three-dimensional?
Is there signaling after SIG gets phosphorylated?
Yeah, is that when the signaling happens?
So SIG phosphorylation you can think, or even PCR phosphorylation by LIN, that's the very first step of signaling. Okay? And then SIG phosphorylation is the second step. And after that it follows to a bunch of other molecules. Well, first you have to have them close together for them to be able to react. Which ones?
Well, Lin and BCR.
Right. But as I was telling you that in a detailed model, we have this clustering, microcluster formations, BCR and Lin can come together, but in a simplified model, we do not have such spatial separation. So, but still you can create a rule which will prevent activation in resting cells. So what could be like a simple rule which will prevent BCR getting phosphorylated?
Well, that Lin can only phosphorylate it if it's bound to the antigen.
Right. That's the simplest choice, and that's what we did. That BCR can bind to antigen only if, or BCR can get phosphorylated from Lin only if it binds to antigen. Now this can happen two ways. You can create the rule in two ways. One way is BCR needs to bind to antigen, and once this antigen binding happens, you create a flag. Flag goes up, and no matter whether BCR detaches from antigen or not, the flag stays up. And that way you know that those BCRs already bound to antigen, so you can now phosphorylate. The other, what will be the second possibility?
They have to be in a complex.
Correct. You follow? So one possibility is that BCR binds antigen, and you set a flag. The flag goes up. That means it has bound to an antigen sometime. That means once it binds antigen, it's ready for phosphorylation, and it doesn't matter whether it stays bound or not. So that's one possible way of creating the rule. The other possibility is it has to stay bound to Lin when it's getting phosphorylated.
You mean antigen?
Antigen. Right. So let me erase this part here. One possibility is binds and now both will prevent activation in resting cells, because in resting cells you do not have ligand. So none of these conditions will get satisfied. So resting cells, you are not going to see activation if I assume these two rules. Now I can have a slightly modified version of Rule 1, which will include this as a special case. It's not quite like true that BCR needs to stay bound, but it's slightly like it will favor binding. Can you think of that rule? So it's not as extreme as this, meaning that BCR needs to stay bound to an antigen. Only then you can phosphorylate by lane. But it's not as easy as like BCR binds antigen, it can ready for phosphorylation.
Do you mean like some temporal restriction? Or if it binds
Some temporal restriction. So you can assign binds antigen for so BCR binds antigen and it does not get phosphorylated or does not have the ability to get phosphorylated right away, but only if you allow a certain amount of time. So it binds and stays bound for a certain amount of time, then it gets phosphorylated. So this rule gives you the flexibility that it doesn't need to stay bound to a light for phosphorylation, but at the same time, it kind of gives more weightage to the binding time. Okay, like if you just allow that BCR binds antigen and it gets phosphorylated right away, that means it basically does not care about the amount of binding or the time of binding. And do you know what determines that time? Like what determines the average time of binding? Like BCR binds to an antigen, right? Ka. Not time, I would say average time and K off is also very important because as you know that when BCR binds, once it binds, what determines how fast it's going to detect? That's K off. So K off is also very important. So the rules we have, BCR binds to antigen. If it's bound for a certain amount of time, there is a flag attached to BCR that goes up. Then lin can bind to BCR and phosphorylate. Once BCR gets phosphorylated, it phosphorylates C. It's as simple as that. And you may ask, like, why we create this like spatial model? Why not use a stochastic simulation? And actually, you can ask both the questions, like why use, you'll see gradually the answers to these. Approach means correct modeling approach. Of course, when we are doing this more detailed model of BCR lipid lean clustering, then you need to do Monte Carlo. But when you are doing this kind of very simple model, the question you might ask that why spatial and also why stochastic. Why not write a bunch of differential equations? So this question you should have in the back of your mind, and you will see that why we need stochastic model and also spatial details.
When they say using names, they don't have so they don't do nothing. They have. I don't know.
I watched that video. That's the guy. She's saying like, yeah, she's saying like all the boss girls. The name was in there. Yeah. That's pretty funny.
Now, I'll come back to the answer to these questions later. And you'll see that this affinity discrimination problem is best resolved when you do a stochastic simulation. Now, to start with, suppose we want to create a stochastic spatial model. Like how you can generalize a simulation or develop a simulation. For example, what you did in homework 1, problem 2, right? You have a 1D lattice and you can go left and right from Now, if you are familiar with such random walk simulations, and that's, I was also, like, I started doing the simulation in terms of these probabilities because I had background in this kind of random walk diffusion simulation and kinetic Monte Carlo. So my first kind of thought was, why not just try to develop a model with such probability-based rules rather than going to energy-based model. Okay. So now, if you have this kind of random walk model, what else do you need? Like, for example, let's forget about lean and 6. Suppose BCR antigen binding. Can you generalize this simulation to create a model of BCR antigen binding? That's the first step, right? So can you create a model of BCR antigen binding using such rules? I can think of like how do you extend this simple model to a model of BCR antigen binding?
First, it would be two-dimensional lattice.
Correct. So one is you need to go from 1D to 2D lattice.
You need two 2D lattice.
Two 2D lattice. So one you need to have place a bunch of molecules, like BCR molecules on this surface, and place a bunch of. Now, in terms of diffusion, the only difference that's going to happen, instead of two directions, in 2D lattice you have 4 possible directions. That's the only difference you will see. Instead of 2 possible moves, you have 4 possible directions to diffuse. Okay. So that's kind of, so now you create a model of B-cell receptor diffusion on B-cell surface and diffusion of antigens on antigen-presenting cell surface. So you created 2 separate surfaces, and you can create a diffusion model for B-cell receptors and antigens. What's left?
If you have the 2 surfaces and you want them to react, then they have to correspond on the lattice.
So the next step is, so this is the diffusion part, which is very straightforward generalization of problem 2. And then you need to have some rules of and there are 2 possible reactions, as I discussed earlier. You can have binding, dissociation. So you have 2 free molecules and bind, or if you are complex, it can dissociate. Okay. And similar to this right-left probabilities, what should come to your mind? That you must have some probability of this binding and some probability of dissociation. Okay. So we have 2 surfaces where B-cell receptors and antigens can be fused on 2 separate surfaces, and then the receptor and ligand can bind with some probability on, and then receptor ligand bond can dissociate into free molecules with some probability off. And one additional constraint, which is kind of obvious, is that this reaction happens when you have like one free B-cell receptor on the opposite side of a free ligand. So it cannot happen, the B-cell receptor is here and antigen is there. Like they are spatially separated on opposite surfaces, it's not going to bind. So once by diffusion, they like on the opposite like surface with the same coordinate, then only this reaction can happen. So there need to be some, now you can create more complicated groups. For example, a BCR, you check the opposite surface and say within some radius R, if you find the ligand, you can bind. So you can create that model. But we do the simplest rule. Only when their coordinates match, you bind. Okay, that means 2 molecules are exactly on the opposite side, at the same location. Then I do the binding. So how does the simulation proceed? I create 2 lattice, 2D lattices, place BCR molecules and antigen molecules randomly on those 2 surfaces. Then pick a molecule. It could be either B-cell receptor BCR or it could be antigen. And then I allow 2 possible moves for those molecules. So I pick a molecule randomly. means it would be diffusion or and then this moves you accept or reject with
So every time you choose a molecule to move, is that a new time step?
so that could be one way of doing a simulation.
It's like a lot of molecules move at once.
Correct, but if you choose that delta t sufficiently small, then it doesn't matter. Okay? But the way we do the simulation is we choose delta t not that small, but in one delta t, one Monte Carlo step, we try to attempt, say if you have 100 BCRs and 100 antigens, we attempt 200 moves in one time step. So in general, if there are n molecules in total, n number of BCR plus antigen, so in each time step delta t, we attempt n moves. Now one thing you need to be careful about, that we do not move them sequentially, which I think I have discussed once, right? That you don't do like, okay, I have 100 BCRs, pick BCR 1, try to do diffusion reaction. Let's go to BCR 2, try to do diffusion reaction. Let's go to BCR 3. It's not like that. Monte Carlo means you randomly pick up molecules. So it may happen that in one time step you pick BCR number 10 5 times, but did not pick BCR number 1 at all. Okay?
Is it only the BCRs that move?
No, both.
Okay.
Both molecules.
So each time you've got to move the BCR and then that's gone. Yeah. So both the BCRs and the agents.
Correct. And if they're in a complex form, then they move together.
What do you mean it's complex?
If they're complex,
yeah.
if they are in bound form, of course you can have this reaction. Or if you want to do diffusion, you need to diffuse them on both surfaces together.
But when you do a complex, do you change the number of moves you do? Do you kind of restrict?
Okay, yes. So that, the best thing to do is to change the number of molecules, to update the number of molecules.
I don't understand what you're saying. When they're in a complex and you choose diffusion, you asked, do you move a shorter distance?
No, so when you're in a complex, you reduce the number of moves you're going to do in the whole time step.
Oh,
You're
right,
reducing the
because
number
you're
of
moving
molecules.
two in the same place.
Yeah.
Is it the probability of diffusion different for the complex than for
Correct. So that's what we assume. And the reason behind that is, this is, as I was telling you, it's a very simplified model of cell surface. Cell surface has lots of structures like active cytoskeleton inside and then other big protein molecules. So typically, small molecules or free molecules moves at a much faster rate than bigger molecules. Okay. And we also have the constraints like no two molecules can be on the same site. So there is a mutual exclusion rule. Now mutual exclusion rule means this lattice, how fine I create the lattice. What's the spacing like delta X or delta L? So that's now set by kind of typical size of protein molecules, like 10 nanometers. Okay. You see, if I say delta X is 100 nanometer, then it does not make any sense to have mutual exclusion. Because if your lattice is so large, how can you have mutual exclusion? But if your delta X is such that it's typical size of protein molecule, then you can say, okay, no two molecules can be on the same site. Now, if that is the case, now you can see when you are moving a complex, it has, like, when you are moving a free receptor, it just checks whether any sites are empty in the neighboring sites. But when you are moving a complex, both surfaces need to have these empty spots where you are diffusing. Okay. So that also slows the complex diffusion. But but that's not enough. We have intrinsic PDIF, the pre-diffusion, different for free versus complex molecules. And that's because this is an effective model and there are lots of other things, and typically complex molecules diffuse at a slower space. So you see, we create these simple rules, and based on these probability functions, you can have diffusion and reaction. And that type of model we call kinetic Monte Carlo. And you can see the difference with what I discussed in the last lecture, that accept and reject moves now not in terms of energy function, but in terms of probability P. Okay. So last week I was discussing you accept or reject a move based on this metropolis scheme or other energy-based scheme, but now I'm doing the rejection or acceptance based on the probability function P. But later I will show you that P has some energy consideration inbuilt, but I'll come to that later. The next thing I'm going to do is, okay, I can kind of create my simulation where I have B cell receptor binding to antigen, and they can diffuse and they can bind with some probabilities P-diff, P-on, P-off. Now what will be, like, okay, I can tell you to do a simulation. What will be the problem? What will be the first problem you're going to encounter? Even if you know the rules, what's going to prevent you from doing the actual simulation? So the first step,
assuming we know P on, P off,
right, those values. So the first step of any computational modeling is what I discussed just now, is that you need to create, even if you're doing a simplified model, you need to have rules which are physical. For example, you cannot create a model where you see activation in testing cells. So you need to create rules which are meaningful. That's the first step, and kind of choose the most important interactions or most important molecules. That's the first step. The second step is to create the model, like you have a bunch of parameters, P-diff, P-on, P-off, and then parameters like what's the number of BCR molecules, what's the number of antigen molecules. Okay? So the second step of the modeling is to choose correct parameter values. Okay? So that we are going to discuss in the next lecture, that how to choose the correct concentrations. Concentrations is easier if you know the typical number of molecules on a B cell surface, and you know the radius of G cell. You just divide by the area and you get concentration. Okay. So that's not that difficult, but the difficult part is to estimate P on, P off, and PTF, and the time scale. So this is delta X. So that will be the topic of next week's discussion. So what I'm going to do today is talk for 5 minutes, and then I'm going to discuss homework problems or any questions on homework.
But it's not going to
You're on your way.
happiness, the inertial score.
Oh yeah.
Yeah, I like that. And this,
Yeah, and then placement of buildings, and
Yeah.
then the other tab is
Yeah,
the
that's nice.
protection at the top.
I don't even notice it. But yeah, I do. Like when you're going.
Why?
You can just stop it.
And then being able to open the wilderness, select the properties tab.
I think it's moving around all the way there. It's everything. It's better than I am.
Label features in the layer.
It is this
The label field should be made.
Oh.
And then
Actually, this
we'll make the text symbol wilderness that italic. And placement properties on so then you go into different properties. Make label be high. This. And feature will be there. On the placement tab, check the removal.
Well, something. Not even. Expand your horizons. That might be more. And something I'm most interested in. Why typically, anytime we have some sort of problem thinking class, I end up being usually wilding by the miles. Oh, what? Circulating to results? Yeah, so that's what interests me. Not hard. This is more that's something. Where it should be fun, right? It is. Still working. I'm frustrated with trying to
Like, let's say you do something.
yeah, the end of the last quarter was so straight. I was trying to go in those two. Just
So essentially the whole quarter you get those papers you can.
not the whole, but you know.
Yeah. Really?
Towards the end, the one I did. It wasn't really worth it.
No.
One of them I couldn't do. But I still maintain ridiculous notation. MOL for mall.
Yeah, oh, no, it's on the second?
Second, Monday. Is it okay? Do you like it's
Wednesday would be better for me.
Sorry. I need to have you act on
is this better?
Wednesday better.
When is the midterm?
Yes, then Monday.
Wednesday's better.
Okay. Wednesday, but not later than that.
Okay.
I'll send an email, and also I think I need to send you this ENN value, the expression of ENN.
you can find the
Problem model.
It should be fine.
So you're really randomly choose a wall, I mean, it's each of them. It's like, yeah. Like how like this one is saying randomly choose a wall. That's better.
Yeah.
So we have to do that in random wall. One molecule.
Isn't it just one molecule? Then there is no like you just choose one molecule. Same molecule you choose.
Yeah. Okay. So it's just so sometimes it's like when he's doing one molecule 1,000 times steps and he's doing like 500 runs. You have to like sort of follow the greatest goal.
There's
Three.
Five.
yeah. But then there's the reasons. And then regular.
Oh, yeah.
They use it everywhere.
There's ones that I've seen.
So that would work.
That one is good.
It's okay.
Just one word doesn't know.
Oh, yeah. Or just that it's surprising someone that doesn't know.
Living proteins.
Because you have to write previous reference.
Oh,
There we go.
some proteins. I put proteins in. If you put protein,
Just about you.
I was like, that
There's only one type of person.
Yes. Can't have plural. I don't think it was just another religion. Yeah, I'd get really excited for that.
You know, it doesn't have nuclear cytoplasm.
Wow.
I know.
Nuclear cytoplasm.
See, that even shows that.
That there's like a biological dictionary so just add to where
That'd be nice.
I guess
No, yeah, that's a word. It's right here, look at this.
the current
Yeah, I believe it.
There's even a type of virus.
He's done those
any
cytoplasm.
changes that you do.
you can add a medical dictionary to word.
Yeah, so say your iteration. Maybe like
Even a custom dictionary.
if you want to fit it like so N5 minus
Cytosol.
So what are you doing there?
Nope. Not cytosol.
It's not a biology. That should just be in the dictionary. I just want to know a dictionary. Yeah, I couldn't find dictionary. Awesome. It's not gonna have like
Oh, it's words in there.
the 10 million page.
What's annoying is even Word math doesn't use apples built in dictionary.
Nuclear
Yeah. Well, it doesn't even have certified.
No word.
Oh, Word. I see. Yeah, Word.
Or hysteresis.
Yeah.
Regulate.
I've been amazed at the things I can find on the dictionary.
Yeah. I wish Word used it. It's annoying. Yeah.
It's really rather ridiculous that at this point, at this stage, that they don't have an update online. Like,
Yeah.
just to
Or every word in the dictionary. Why should there be any word in it?
yeah.
Yeah. Let's see.
In the Oxford.
Oh, it's not in that? Wow.
Yeah, that was my guess. But there's no excuse. The dictionary is not being online updated and
Has certified? Oh.
Yeah.
what is this, 1998?
It still updates. What are you buying? No, it's like 2003. Look, it doesn't have a squared logo. That's
Right,
good.
that's when they realized that aesthetics is something.
Yeah, exactly. So it's post XP.
Those macro.
Yeah. So after 2000.
Oh, I see. So bubbly.
2004.
That's like, this is the edit.
That's a what?
This is like the XP. See it art.
Yeah.
Look, we can do curves too.
It's version 11.5.7.
Okay, thanks.
11 versions of it since 2004. It's more than one version a year.
Yeah, I wonder how many were. Yeah.
It's the first quote that I think he's left to
Would you make that
Side, left hand side.
Do you guys have the homework problem completed? Do you know how to do 2A, estimating the probability distribution PR? Do you know how to do that?
You just store the random numbers that you generated and then plot
Right.
the histogram.
Histogram. So and you do that at time say t1. How do you do that? So you do the Monte Carlo simulation, and after some time t1, let's say t1 is 100. When e equals 100, you calculate what's R. So R is R1. So this is from first run of your simulation. Then you do another run to at t equals 100, you find position is R2. The third run, suppose you have R3. So for each run, you store these numbers at t equals 100. Okay. have this set of 100 numbers stored in a file, and then the command in MATLAB is called hist, and whatever the file where you store this data, hist of this data, that will create a histogram for you. And that's the probability distribution. And what probability distribution means, in the x-axis you have R, and the y-axis is number of runs. So for example, out of these 100 numbers, you have R equals R between 0 and 10, 15 times. So that means here, between 0 and 10, you have a bar of size 15. So histogram means on the y-axis is number of runs or frequency. How many times you get this value. Okay. That's your y-axis. And x-axis is the position. So you create, this is t equals 100, so I should not, let's have say 1,000 runs. So this is the probability distribution or histogram at t equals 100. The more you increase the number of runs, the better looking histogram you have. Okay. So if you run the simulation 1,000 or 10,000 times, your histogram looks much better. Like a nice car. If you run it 1,000 times, versus if you do it 10 times, your histogram will be like this. So if you increase the number of runs, you get a better shaped curve. Mimics the actual situation.
So to get closer to that value of PR, we calculate part 1.
You calculate, sorry?
You know, the value PN, and we calculated part 1, we should get close to that.
Correct. I was going to tell you that. Yes, exactly. So this PR, if you have large enough number of runs, should be and n is the time, correct? So here you can say this. So the expression you get E and N in problem 1, the probability distribution in problem 2 should match that for various time points. Random walk is one problem which is simple enough to allow you to do both exact solution and two simulation.
equations or the
it's up to you. It's up to you what you want to set up. So discrete time will be similar to problem 2. I think in class I wrote continuous time. Correct? And I did like this two-stage system in the last lecture. Did I write a continuous time equation? You understand the difference between two, like the continuous time and discrete time. So when you do,
so with the discrete time, you'd solve it using the GSN.
For this problem, you don't need to use GSN.
Okay.
It's simpler. It's much simpler. Yeah, I should have told you. Yeah, it's the reason you don't need to do GSN is. So when you are doing the random walk master equation in problem 1, you have essentially, if the problem is defined on an infinite 1D lattice, you have essentially infinite number of possible states. Okay? Whereas in this problem, this is problem 1 and problem 2, you have only 2 states. So you just go between 2 states. That's why the problem is much simpler. You can do exact solution. It doesn't matter whether you write it in discrete time or continuous time. The only thing you need to remember that the probability. So master equation is always in terms of probability. So here, probability of finding at site n versus probability of finding at 1. Right? So that's how the master equation for random walk is written. Here, the probability of state E0 and probability of state E1. And basically, you have one independent variable in the system because P0 plus P1 plus 1. Okay. The system can be either in this state or in the E1 state. So the probability of finding 0 plus probability of finding 1 is 1. So that means you have basically one independent variable in the system. So it kind of turns out to be a one ordinary first order ODE which you can solve if you write it in continuous time. Or if you do discrete time, then you just do like a difference equation, which is also simple to solve. But you don't need to do GSM. And the reason is you just have only one independent variable. Whereas here you have infinite number of independent variables. It's not independent, but they are coupled to the equations. But you have infinite number of possible states. to try for B and C as well? Yes, no, maybe. Last one I was teaching on the course, and I got one comment which I never heard of before, saying that I can't believe that I got an A with so little work. Three. This is three. 3A.
Right click and zoom, yeah. Right click and zoom to layer on counties selector.
Because I just took so when you say, I'm reading of my notes, when you say the chocolates assume that D like 2, 1, so D is like what's D means? Is it like the probability of going down on that D2?
Okay, so D, if you're thinking in terms of continuous time master equation, D is like per unit time, what's the probability change? So D is not probability. So if you look at what I did for this two-stage system. Okay, let me write down.
So D is like probability of changing from 1 to 2.
Correct, but it's not the. It may not be the probability if you write it in continuous time. So let me write down the master equation for random walk, both in discrete time and continuous time. Okay, that will make it clearer. So if you write it in discrete time,
You define n plus 1.
so you see in the continuous time, you have a derivative with respect to time, del P del T. So this is the probability, this is not the probability but the rate, n plus 1 to n. So this is the rate at which you go from n plus 1 to n. And this is the rate at which you go from n minus 1 to n. And then from n to n plus 1 and n to n minus 1. So these are rate constants. Whereas here, if I just generalize the random walk with left, L and R are probabilities. You can also introduce some sort of weighting problem. So when you are writing a discrete time equation, you have discrete steps, and then the coefficients are probabilities. L, R, W, those are probabilities, each less than 1, and they add up to 1, like any probabilities. Whereas here, when you have continuous time, these are rates. These are rate constants. These are not probabilities.
So what would the units of rate constants be?
So rate constants unit will be second inverse, right? So you have P on both sides, which is unitless probability. What's the unit here? So that means P has to be second inverse. And when you write discrete time, these are all unitless, like L, R, W to the probabilities. Basically, you do not have it.
So that's why when there's only two energy states, then it's basically half the number of terms there, so it can only go, like it's in the lower energy state, it can't go lower. So in this problem, there's only two energy states, but it's kind of half of that equation. Like, I mean, you know, just without few of the terms.
Oh, yes.
Yeah.
Correct.
I'm just trying to understand
Yes.
the problem.
Correct.
And then to solve the master equation, I'm not really sure what else we need to involve to solve that.
So this equation, there are 4 terms.
Yeah.
This is from n plus 1 to n, from n minus 1 to n, and then n to n plus 1 and n to n. So now you can just say, okay, this n plus 1 is like state 1, n is like state 0, and then you have this from 0 to 1, and just get rid of the other 2 terms.
Yeah.
Then you basically wrote the correct equation.
So that's our master equation. But we want to solve it for P.
So now you have
Yeah.
an equation in, this is like equation in P1, and you have the same equation in P0, except the signs will be flipped.
Yeah. But like in this question is what we're trying to do, find
Solve.
P1
Right.
T.
Now how do you solve it? So you see this dp1 dp, but now I told you that probabilities must add up. So this P1 and this P0, they are not independent. So you can replace P0 by 1 minus P1. So basically you end up with an equation in P1, which is a first order ordinary differential equation which you can solve.
So what's the steady state equilibrium distribution solution?
So steady state means when the left side is 0,
Yeah, right.